세션 마지막 강의인데요. 아미트 션나 박사님이시고요. 유니버시티 허스피탈 보온에서 지금 독일에서 오셨습니다. 오늘 박사님께서 말씀해 주실 주제는 Cytokine Induced Killer Cell Therapy Engaged Since 30 Years for Getting the Cancer Cure with Leech 라는 타이틀로 재밌게 강의해 주실 것 같습니다. 감사합니다.
To thank the organizers for arranging such an interesting event and also for inviting us to share our thoughts and data about 6 cells. My name is Dr. Amit. I'm working in the Department of Integrated Oncology in Bonn, Germany, and we are very much focused on 6 cell therapy. So, to start with, so you see the picture of Professor Schmidt was the pioneer who has developed this therapy in 1991. During the time when a lot of therapies was coming up, he also came across some preliminary data which he translated in the first clinical trial, and this is how the 6 cell therapy come into this clinical scenario. The basic idea is very simple. You take blood from patient, you expand it, you enrich it, and bring it back in the patient. Theoretically, it looks like this is just immune cells, but it's not so. These cells, when they are infused back in the patients, they are acting as a specialized cell. They obtain the tendency to kill the tumor cells. This is how it has been proved by a lot of clinical trials, and thousands of patients who were benefiting from this. And also, the interesting point was these cells are not toxic and easily compatible with all the cancer therapy protocols. For example, you can combine it with checkpoint inhibitors, epigenetic inhibitors, and also bispecific antibodies are just few cases. So this much reference is enough to go forward. In the next slides, I will try to show you the work what we have done in last less than 2 years. The idea was, can we improve these cells? So, but before that, we also start, should we reassess the data of the clinical trials where the 6 cells was used? So in this last 30 years, a lot has been known. We and other colleagues have tried to understand a lot of things, but we are surprised every time when new and new data is coming up. This picture we have newly generated, and we are now writing an expert opinion article where experts from 6 cell therapy are writing that how much we have understood in the last 3 decades and which parameters are still untouched. So we can expect that next year this article will be online, and I will not go into too much of the details, but briefly I can say till now 2 pathways has been somehow summarized. One is when we introduce the 6 cells, they need to have a direct contact with the cancer cells. So this is how they kill. Second, when they don't need to get contact with the cancer cell, when they suspect that cancer cell is in the neighborhood, they release something. So they release some molecules which are cytotoxic for the cancer. And now we are also working on the third hypothesis. I don't have the data yet, but maybe in the next meetings I will be able to present that when cancer and 6 cells are in the same tumor microenvironment, how cancer detect that these are specialized immune cells. They are just not immune cells. So there is something special happening. We are trying to figure it out. We have a team of doctors and biologists who are trying to reassess the data sets and also planning their experiments according to that. For example, you see the picture of Ying Yang. She recently finished the PhD. She is more interested in the lymphoma. As a medical doctor, before she started her work, she said instead of just running another clinical trial where she just enrolled randomly patients, she would like to see what is the outcome of last successful 17 clinical trials. She summarized her findings. She made a timeline. When was the clinical trials was done in the clinical trial, and what was the information from the preclinical studies. It's a nice article. I would recommend you please read it out. That's why I just took the screenshot of directly from PubMed. I will not go much to the details, but this strategy worked very good for us. Now when the student comments want to start, especially the medical, we ask that please reassess the cancer type you are interested in. So we just do not redo the things, or we just do the boring stuff because patients are dying every day. We need to optimize. We have to do something new. Similarly, this student Raula, he's interested in the lung cancer. He tried to, before starting the experiment, he come up with the information that there is a very less data available for 6 cells and anti-PD-L1 and CTLA4. And now, this was also surprising for us because as I showed in the first slide, there are a lot of clinical trials. When you see 157 clinical trials, you do not do one by one. But when he brought this information to us, it was clear to us that what experiments we have to plan now to fill the gaps. Then we have this Lulu one. He just finished his PhD last month. He is a hematologist by profession. He is based in China, and he had a very noble thought that cancer patients after a certain age, most of them are usually before detecting cancer, people after 45 or something, they usually have diabetes or blood pressure. So patients, or normal even human, they used to take medications, so which are not cancer related. He picked up one medication called Medigreen. This medication is used for the blood pressure. His thought was, when the patient is detected, he's already taking some medications for his physiological processes. Could it be that these medications work or feel compatible with the inhibitors or non-inhibitors which we did in the clinical trials? And how is the scenario for the cancer immunotherapy? And he made a lot of interesting analysis in globin transcriptome or something, and he identified that these non-cancer drugs can also show some anti-cancer potential. This was also surprising for us, and more interestingly, he showed that these non-cancer drugs are compatible with the epigenetic inhibitors. So that means when you have a patient who is taking long-term medications for diabetes or blood pressure, you can reassess that these medications will also have certain impact on their clinical scenario later on. Now, this is $1 million question. As you know, this cannabis is already legalized in USA and in Germany. We are on the process to have it. I mean, I personally do not want that our young generation take cannabis, but somehow biologists has shown a lot of positive things about this. And this Francisca from us, she just as a hobby project, she just wanted to understand what will be the scenario after 10 years when German patients will start taking cannabis. So whether we have to reorchestrate or reorganize our studies or something. She gave us a very first hint that high expression of cannabinoid receptor 2 was on 6 cells. And she checked multiple myeloma, and she was able to show that the CBDs was able to not only decrease the tumor cell, but have a protective role for 6 cells. This was kind of first hint for us, but was not sufficient. Because when you see the picture here, I just put the image here, but there is a lot happening when you use this cannabis. Then we wanted to go further for this one because multiple myeloma is very heterogeneous. The results could vary, especially in these kind of studies. So we focused on lung cancer cells. This paper is in a research square. We are finishing the revision now. Hopefully it will be accepted also, but you can also read it. It is freely available on the web page. So Utah made a lot of experiments to conclude some data, and she confirmed that, like Francisca observed something in multiple myeloma, in lung cancer also, CBD has some potential with the cancer immunotherapy. But the clue was not for everyone. She confirmed that a strong genetic background, for example, the patient who has a mutation in KRAS gene, or patient who has this EML or ALK rearrangement, they will be highly responsive for such kind of treatment. And she also confirmed that it's not finishing up to that you add CBDs it has some molecular processes, 6L has some cytotoxic profiles. Overall, this combination is also activating some certain pathways. We don't know what is the consequence of these pathways because this is kind of new studies that you see that it's not even published yet. But surprisingly, I just asked her as one like request that could it be that it has some epigenetic effects later on? So instead of going for DNA methylation or transcriptome-wise changes, we check for line 1 mRNA. Line 1 is a repeat which comes through the evolution in human, and these repeats can get methylated. So based on the cancer type, the expression of line 1 is also changed. And she confirmed that the effect of CBT in lung cancers, together with or without 6L, was also making some line 1 expression related changes. That means epigenetic was effect. So that's why I said we do not know much the consequences, but there will be certain consequences when we will replicate these findings in the in vivo system. This CBT story gives us another hint that if CBT can influence 6 cells, or if 6 cell influence CBT, or immune checkpoint inhibitor. So what is the combination? Who is predominating? They both work good, but who is predominating? 6 cells, immune checkpoint inhibitors, or CD kind of things. Then we have this Fang. You see that she has a Mickey Mouse kind of thing on the head. She is going to defend on Tuesday her thesis. She is also a hematologist. She wanted to verify the things that if there is a mechanistic approach running behind the function of 6 cells. So she just made a very simple arrangement of her experiment. She took the 6 cells and HSP19 inhibitors, which has been used in the clinical trials. She picked up blood cancers, myeloma, leukemia, and lymphoma. So different types of blood cancer. The experiment was on the same setup, and she was surprised and she confirmed that myeloma and leukemia, there she detected NKG2D mediated pathway. And in Burkitt lymphoma, the same combination was causing fast and pathway. This is very interesting finding for us. It gave us a clue that what we have learned till now, there is still much more to learn. Now, why 6 cells has to go through 2 pathways just with the same kind of inhibitor? Who is guiding this? Is it, we say cancer is clever, but does it mean that when we generated 6 cells, do they also got some kind of flexibility or dynamics that they can also judge how cancer is going to get? So it's like 2 intelligent players are in the same environment. Now, for us, this information has opened many new experiments. We wanted to know also that are there other pathways involved which we have not even touched? Because when you talk about cancer, you go to textbook or you go to other sources, there are certain pathways, for example, BNT pathways change, or similar P53 pathways are changed. So there are limited pathways in which cancer can react. And we was always thinking that there are limited pathways which 6 cells can react. But when they come together, it might be there are still some passive pathways which we don't know yet. And they might be the key to tune the 6 cell for further investigations. While, as I said, we are reassessing a lot of data, Jingjing, he come up with some information. He's working in collaboration with Shanghai Hospital. He said that he's especially interested in histone inhibitors for the epigenetic purpose. He said that histone deacetylase inhibitors and many other combinations has been tested for several clinical trials for multiple myeloma. He also said 6 cell has also been tried for many clinical trials for multiple myeloma, but somehow they was never tested together. So it skipped somehow, it was forgotten instance. Then he come up with the idea, the first thing we ask, does it make sense to check it? And he did a lot of experiment, I'm just showing you part of this. His 2 papers has been accepted, and this will be the 3rd one, showing that histone deacetylase inhibitor, especially HDAC6, which is kind of very specific and very important compared to other palm cancer HDAC inhibitors, he is going to suggest that HDAC6 inhibitor to see in which subgroup this combination will work with more efficiency. So he wanted to propose before any clinical trial which subgroup of patient will get maximum benefit from this. So it will reduce the work because in the clinical trial you put data from all the patients, you hardly care for genetic background, you don't describe much male, female or grade type, just in the graph you see significance, non-significance, something like this. But his thought is, in his next clinical trial he will focus on the subgroup which he will prove here that which will get maximum benefit. So as I said, we are trying to still understand the mechanisms. We wanted to tune these cells. We wanted that these cells, when we infuse them in the patient, they live longer than they are working till now. We think that if the cells can live a little bit more longer, they will not, first when they enter, they might release something to kill the cancer cell, and then they find the cancer cell, they make a direct interactions. But probably the time is not sufficient for them to work. And if they will live longer a little bit, we think the results will be very much significant. So we are trying to understand more. I am just putting here 2 papers that it is beyond synergetical effect. We think the strongest point we have is 6 cells are not toxic. So if something is not toxic, it's just taking a little bit longer, that means you can tune it. And if you get success in tuning them, the results will be, of course, interesting. This slide is very important for us. We have written this article recently because all the therapies which are based on the T cells, they admit or they don't admit, they have a problem with T cell exhaustion. It's like those who knows this, I put some points here that when the T cells get tired, they cannot work. It's like an employee or colleague who is tired and is not able to work properly. You suggest, okay, take a break or something, or take a holidays or something, come back with more fresh energy. But in therapies where excessive T cells are used, they cannot do this. They are still trying to implement it. But in case of 6 cells, we have advantage because we have heterogeneous population of cells. So if T cells are tired, we still have NKT cells. So there will be a subgroup, subpopulations which will work, still keep on working, although slower than the normal, but they will still keep on working. This is the new thing we are trying to explore now. First, we wanted to know actually how much exhaustion we have with the 6 cells when we make them ready for the experiments. Second, we don't need to reverse it, but can we tune that instead of, for example, certain percentage is getting tired, but we make them a little bit lower? Or should we make some modifications in the protocol, light modification that the T cells don't need to get tired or something? In other words, if I say that all these technologies which take PBMC and then they specialize the cells, CAR T cells or 6 cells or something, these cells are cultured in artificial condition for a certain amount of time. For example, for 6 cells, you need to have these cells cultured for 2 weeks. I think CAR T is 3 weeks. So after taking from the human, these cells are artificially trained. So are they biologically and physiologically young enough? We don't know, and nobody is actually discussing that. But we would like to answer this question through this mechanism. We would like to show that after taking blood from a human, after and before preparing it back for the transfusion, how they are. Are they biologically young? If they are already biologically old, so then we don't need to bother because that's why they don't live so long. But if they are young, that means they have a chance to live longer in the human body. So as the title shows, that it's not about the competition of different therapies. We still believe that therapies can complement each other. We recently wrote this article at 30 years of 6 cells, and we showed that the field is advancing at a different level. Now we have DC6 cells, we have CAR6 cells, we have DC6 NK cells. So these further modifications of 6 cell therapy is a proof that we cannot ignore this therapy because it's safe. We don't have a problem of toxicity here. Of course, there are some minor side effects, but till now, all the clinical trials which has published on the people we have talked, they always claim that these side effects can be handled. They are not something very severe. So now this modification gives us a hope that we will understand more how these cells are working in the body. So, we have initiated something. As I said, this therapy is three decades old, and this year in August, you can see here, we arranged this first conference of sick cell therapy. We invited all the people who was working in the field, are still working, and we arranged a good number of speakers. We could not arrange USA and Canada because of time differences, and speakers was sleeping at that time. But somehow we arranged it, and now we made a network of people who are in this field, and they all appreciated it because this thing was missing. We openly discussed some things, that how we have to restructure ourselves. The idea was to meet every year, virtually or with hybrid talk or personal meetings. So this year it was in Germany, so next year, in the middle of next year, the meeting will be in Italy. And of course, it will be advertised and you all will be informed and you will be welcome. And now the community decided to make an International Society of Sick Cells. So this society would like to support basic research, translational research and clinical research. And also they would like to make a guidelines. Like for example, when the methodology to generate sick cells we would like to have a website where different people can show their methodology, or they prepare the sick cells so people can decide how much are the differences or similarity in the protocols. Then the guidelines for the clinical trials, so when they introduce the cells, how much cells was used, what was the grade, which was the markers they have used time to time, and how long the therapy was followed, and if the patients was also checked later on after they was freed from hospital. So these all things will be organized, and Professor Schmidwalk is now the first president of this society. So I also say to you people, if you are interested in cancer immunotherapy in general, you can be a part of this society. There is no cost for that. It will be handled from Germany, and we will make a network of clinicians, researchers, and students. And we will have also one section where people can post their comments, post their questions. And the expert will answer it, so everyone can see, okay, so did we miss something, did we overlook something, can we improve something? So these are the criteria we are following now. Overall, we think that with this networking connections now, we will be able to perform better. Because for a single lab, to have an idea, to test it in vitro, then to go to in vivo, and then to go to clinical trial takes longer. But in this network, we will be able to distribute our work. The people who has in vitro possibility, do it. Who has in vivo, please replicate the studies. And the clinical people, they can easily start making paperwork for the future clinical trials. And very open discussion, we would like to have very transparent scenario for the sick cell therapy for the future. So this is my last slide. So as you see that we have a team of PhD students and medical students who are working hard on that. I just showed some of the studies we have done. But if you check in the PubMed, we have done several new work. Because of the time strain for 20 minutes, I didn't want to go to the details for the graphs or something. And we have collaboration, in-house collaborators also who are willing to participate with us, because from Korea, recently glioblastoma clinical trial was published in the sick cells. And we had a very interesting discussion in our campus that this is nice, that how sick cells actually, peripheral sick cells, how they could communicate with the central nervous system immune cells. And we are also planning something in this direction. And then we have all over the world now collaborations, and of course the colleagues who are working with the sick cells all over the world, they are the part of this network. We are funded by the German fundings, Bone for and European funding. So, as this picture shows clearly that I am from the city Bonn, it's nice to be there when it's summertime or Christmas time, so you all are welcome whenever you have holiday time, and thank you very much.